all yours.
Great. All right, folks, so we're going to have a little bit of a round table discussion or a conversation with the audience. The topic that we thought would probably be of most interest was around the concept of the in vivo testing of antivenoms and other products with living animals, mice in particular, and how the 3 R's should apply, what are the alternatives, what are people's perspectives on the future of this sort of work. And so to start, I might ask Professor Calvete to give us some short comments with his perspective, and then we'll hear from David Worrell, and then we'll have some questions from the audience, and some questions and answers from the panel members. So Juan, I'll give you a couple of minutes to make your case.
Okay, how does this work? So I will make a statement before starting the panel. I don't have any joy killing an animal, but I have joy eating a cow. So the things are not white or black. So being in favor of killing animals, of using animals in research, is not that you are always against or always in favor. So for me, a discussion is like a chess play. You make a move, this is your arguments, the other party move and give arguments and so on. So if someone convinces me that we have reliable alternative that give reproducible results that can be used for regulating the products, and that all agree in all stages of the product, because it's not sense that you use something that cannot be used in the practice. If this is the case, I have an open mind to change to any alternative that meets this criteria. My personal view is that we are not at this stage, and this is, well, I will try to put arguments to demonstrate this, but if you can convince me of the contrary.
What was your argument?
My argument.
Yeah, you haven't yet told us why they're indispensable.
No, I said that my argument is that currently I don't think we are in the stage of finding alternatives for today's replacement of animal testing by any other kind of testing.
Okay, but specifically why? What is it about the mouse model that can't be easily replaced?
It's physiology. So it's a living animal, and you are using for testing products that will be given to another living organism. And so you try to reproduce the pathology in a living organism to reverse the signs and symptoms with a drug, whatever drug is. And this has consequences for the animal, and the regulation, at least the ethical regulation, says at the end of the testing, you should kill the animal. Why? You can let it also go on the street or whatever, but the regulations are also have to have also into consideration. So in principle, I have everything I have read and searched for alternative for this physiology. You can use another animal, a rabbit or even a sheep or whatever. But the most convenient one, the most that you can more from an economical point of view, for the easy to raise them in a proper condition and more humane conditions. So the mice in an animal house, they live better than many humans in many places of the world. So they are raised for this. They have a purpose. The purpose is to do this proper testing. And if you do a proper testing, you need to do it once. If you do it not properly and with not statistically strong testing, then you need to repeat, and people need to repeat in its facility one time and another, put under different conditions. So at the end, you are wasting more animal lives than if you do proper testing. So I'm for the proper testing if there is no a proper replacement. And I guess that for living organisms, there is no in vitro testing, not ex vivo testing that can replace them. But this is my argumentation.
Okay. David Worrell, how would you respond?
Well, my concern is not killing an animal, euthanizing an animal. It's causing an animal to suffer. And I fully accept that in the pursuit of improving human health over the centuries, perhaps, and continuing in the present, animals have suffered in the interests that we can improve human health. But I think there's a limit to the amount of suffering. I don't believe that any degree of animal suffering is justified just because it's the interests of human health. I think the balance between human rights and animal rights has changed over the centuries. And I think that my concern with in vivo testing is not the anesthetized animal, but the animal without analgesia that is injected with venoms. And one of the most common characteristics of all venoms is that they produce pain. And I've been bitten by a snake, and I realized that for the first time how stoical my patients have been over many years. In fact, it can be intensely painful. Of course, some snake venoms are specifically and notoriously agonizingly painful. Some of the coral snake venoms, for example. So, just to summarize, certainly animals have to suffer in the interests of human health, but there's a limit to that suffering. And my frustration is that there hasn't been much response in the toxinology community to decreasing that pain. Now, John Harris, Rob Harrison, and Chema Gutierrez all investigated the using analgesia, not anesthesia, analgesia, routinely for mice involved in experiments, and they demonstrated that this doesn't necessarily alter the thresholds, the results. So why hasn't that been adopted? So my final comment is, look, if the toxinology community doesn't do something about this, it will be taken out of our hands. There will be a public reaction, probably a hysterical and exaggerated and inappropriate reaction, which will nonetheless threaten to bring the whole of our speciality to a standstill. So I hope that's clear. Not about killing, it's about animal suffering. It's about a limit to what is an acceptable suffering for an animal if the outcome is to improve human health. And why aren't we adopting means of reducing this suffering? Not just one or two isolated outposts like Liverpool, but generally throughout the toxinological community.
Okay, so Andreas, you've got another lab that does a fair bit of work with rodents. What are your perspectives, and how does your lab work to try and minimise some of these problems and comply with all of the 3R principles and other regulatory and ethical requirements?
Yeah, so thanks. First of all, just to clarify, my lab doesn't actually do any rodent experiments ourselves, but we work with collaborators, so we are, you could say, of course, co-responsible for ensuring that we follow ethical guidelines and so on, but we don't actually do it in-house. So I do speak a little bit on behalf maybe also of the collaborators that are doing the experiments. I agree pretty much with everything that has been said so far. I think we try, of course, not to do any unnecessary animal experiments. Also simply just it's very costly. So even if we disregard ethics, it's super complicated and costly. One thing is the animal itself, but also making materials, shipping it elsewhere. And there's a lot of variation in animal. There can be animal experiment. Well, it's really expensive. Juan is shaking his head, but making a lot of nanobodies or antibodies for testing is the biggest pain we have when it comes to financial trouble, just because it's a lot of labor. So we try to minimize it for that reason alone, of course. We try to think about when do we have a sufficiently large sample size so that we can say something with statistical significance, because I agree very much with your point, Juan, that doing something only half well or with not enough so that it's inconclusive, well, then you might just have wasted, and even worse if you conclude something strongly and have strong claims and others read your work. And I think there's been some examples I can think of in the literature where maybe very strong claims are being made. Others will just assume, well, those claims are probably correct, and they will continue working maybe with molecules or models and then end up wasting even more resources and animal lives down the line. This is particularly the case for, you could say, my field of antibody technologies, where there's some extremely, I would say, hardcore anti-animal experiment people that want all antibodies to be completely animal-free. It may never have been introduced into a mouse. But the result is that if you create reagents like that, you end up having faulty experiments down the line, and you send the wrong molecules into in vivo testing. So potentially by saving one mouse's life, you end up killing 10,000 unnecessarily. So I like the aspect that you said, Professor Worrell, with seeing what can we do of relatively simple means to reduce suffering. I'm not an expert on what can be done in labs. I know that there's, in some cases, a little bit of an issue with sedating animals if you want to look at neurotoxic effects, and there's also been issues that we would have run into if we followed some of the guidelines that I know exist at Liverpool, where you kill animals early on to avoid extended suffering. Fully understandable, but I know with some of the work we did in Costa Rica, we would not have uncovered that dendrotoxins are actually not that lethal if we had killed the mice when they looked like they were going to die. So again, it's not a black and white discussion. I think it's important that you try, both for ethical and financial and many other reasons, to reduce and replace animal experimentation, but you have to be very careful about it. You have to be careful whether your new model is representative enough, and you also need to, which I think can be a bit of an issue in this field, that if you've studied regular antivenoms, serum-derived antivenoms, they sort of behave very similarly. Maybe at least the fat ones can be compared to fat ones, and the fat 2 can be compared to fat 2, and IgGs can be compared to IgGs. But if you suddenly go in and make a completely different type of molecule, an aptamer or a small molecule, even a monoclonal antibody, the behavior can be quite different. So using some of the models that are okay for qualifying antivenoms or testing them cannot necessarily just be used for completely new modalities. And I see quite a few papers out there where people pre-incubate things, they stick it into a mouse and they conclude, wow, it works. But I mean, that would also be the case with sodium hydroxide. And of course, it's maybe the starting experiment to use those pre-incubation models, and if it doesn't work there, of course, then it's a bad sign. But we uncovered an antibody that worked fantastic when it was mixed and injected into a mouse, but when you gave it in a rescue setting, it actually enhanced toxicity. That is not something I can imagine we would have found out only using in vitro methods. So, you know, it would be good if we could reduce animal experimentation, maybe even completely get rid of it for antivenom manufacture, and then for experimentation, we can, you know, think carefully about how do we limit it, but not over-limiting it so much that our science ends up being bad. And one last comment is that I think our real struggle is actually not internally in the, you could say, amongst us scientists. It's very much journal editors that demand. They have an unlimited hunger for dead mice. some of the journal editors, and I don't know what to do about it. They are not convinced when you tell them that actually this pseudo assay is a great replacement. They just don't buy that.
So Anna Maria, from Brazilian perspective, Tantan, what are your
okay, I can say a little bit what we do in Butantan. For me, the biggest problem is still making antivenom in animals. We have a lot of questioning about that. In what we do about that in Brazil, I think our horses are very well kept. So the farm is, we used to say it's a 5-star hotel. What we do in the lab, we have a very strong ethical community. We still can do, we can do animal work, but we have to control the numbers, and so we work as the best possible with the 3 R's. And I think the reducing, we avoid to repeat experiments and all of that. We don't use analgesic, but I think it's a good thing to discuss in there. And what worries me a lot is the last thing that David said. In a short time, we probably will not be able to use animals anymore. And in this regard, I have some concerns about what we are doing. So it's to be completely independent. Because, for example, about antivenom production, I think what is being doing is great. I see, but we still have some answers for elapid venoms. We cannot, we don't have any good answer for viper venoms, which are more complex. And again, how long will it take until we could make it in affordable prices? So this is, for me, a good task for the scientists, is to hurry it up. For example, now in Butantan, I'm pushing them, okay, we need to change the production and blah, blah, blah. This is one thing. And the other thing is the lab work. I 100% agree with Juan. It's very difficult. I see a lot of models. For example, if you use cell cultures, it's wonderful to see myotoxicity. You can do immoblasts. If you want to see action on keratinocytes and tissue cultures, organoids. So we have a lot of models, but none of them replace the complexity of the live animal. So in the end of the day, if you want to see if this inhibitor protects against venom, you need a more complex atmosphere. Otherwise, you will do some clinical trials with something that I think the preclinical were not correctly run. So again, what's my position about the scientists together here is try to think about venom as a complex stuff. So if you are working with one cell culture, try to mix the experiments, try to join. Like I was discussing with this morning, try to think about the other components that play in the local lesion, especially for viper venoms again. So we need to have nice models, and nice models now is only the mouse. And I hope we can make this change as soon as possible. But by now, I agree with Juan, we still don't have how to replace. And thank you.
Okay, so Nick, your turn. So you guys have done some work with 3Rs, fairly active in that. From, say, the way things work here in the UK, you know, what are the complexity of the problems that you face when you're wanting to find a model that you'll actually be allowed to use, and what are the constraints?
Yeah, I mean, it's a good point. I think we are constrained in some ways in terms of the flexibility, the duration of the model, the severity of the model, and everything is very heavily regulated. And you have to make a very compelling argument for the nature of the experiment. And we're talking here about severe rated experiments, the most severe classification of animal experimentation in the UK. And often we're talking about large numbers of animals, particularly in the context of batch testing antivenoms or developing antivenoms. And so it's becoming more and more difficult to make that case compelling. But I think ultimately what the regulator wants to see is they want to see your arguments around the 3 Rs and implement things that are going to enable that work to be justified. And if you don't meet those thresholds, then you will not be allowed to do the experiment. So, I mean, we started with replacement. I think Juan described that at this point we don't have a replacement model for evaluating therapeutics for snakebite, and I think that is true. We don't have a single model that accurately recapitulates the in vivo environment of a mouse. To your point, David, there is research going on to develop new models, still in vivo models, in insensate hen's eggs, for example. Work is still being developed there. We have researchers who have been active in that space in the room with us here. Galeria, zebrafish, people are actively looking at alternatives. Whether they'll replace a rodent model remains to be seen. But I think the key here is in the meantime is what can we do to both reduce and refine those procedures to make them much more acceptable. So reduction, I think, is an obvious one. How do we reduce animal numbers? How do we reduce the extent of suffering, the severity of envenoming that we're imposing on experimental animals? And I think there are ways to do this. The frequency with which antivenoms are often tested could be looked at. Some manufacturers do test every single batch they release in a preclinical model, and there are ways to standardize antivenom production perhaps to reduce that. We can also reduce experimental numbers in our own experiments depending on how we design those experiments to answer what particular question we want to do. And I think we can robustly reduce the duration in some cases as well, as long as we validate the outcomes of those experiments and ensure they're going to be reflective of what we're doing. And then on to refinements. There are clearly refinements. Analgesia is an obvious one that can be implemented. We have now routinely been using analgesia for all of our in vivo research for the past couple of years to again reduce the suffering on experimental animals, as well as how we perform experiments. So if we were performing a concentration dependent experiment like an LD50 or an ED50 experiment, rather than just preparing all of our doses and injecting all of these mice with all of the venom, not knowing what's going to happen, we will stage the experiment. So we'll dose one group of mice, we'll see what happens, what's the response, then we'll adjust our dose. Do we go up, do we go down, and monitor the response and continue in that way. And that's been really successful at reducing the number of animals that we've used in these experiments over the years, and we've kind of been able to show to the regulator as well that reduction in the severity and envenoming of the preclinical work as well. So I think there are things that we can do. I totally agree that there needs to be increased research in this space. I know that Stuart Ainsworth at the University of Liverpool has been developing new models of snakebite envenoming still in an in vivo setting, but reducing the severity of envenoming such that you don't get a severe rated procedure, and trying to make it more reflective of real-world envenoming too. So there is at least some research in this space. But I think we also have to be aware that most therapeutics for snakebite are not going through a traditional clinical trial process. And this does in some way make preclinical testing more important than it can be for other medications. And so I think it's going to be, at least for the short term, medium term, it's still going to be an absolute requirement to perform in vivo research to validate therapeutics, whether conventional or antibodies or drugs or whatever they might be.
Okay, so from a regulatory perspective, it's very interesting because The major problem that manufacturers have, of course, is that they have to test according to what the regulator requires. And in most countries, the requirement is what's in the pharmacopoeia. So if the murine LD50 and ED50 tests are what are in your pharmacopoeia and you don't deliver those, your product won't get approval for release. So you're stuck with that. Now, practically speaking, if you have an alternative, there's quite a lengthy process of validating that method across a number of different national control laboratories typically to determine that it's going to be reproducible before you can convince someone like, say, the Expert Committee on Biological Standardisation at WHO to put it forward. But having said that, just recently we've had a white paper out from ECBS on the use of animals in testing everything basically but antivenoms. And the idea is that they want to do away with the use of mice for everything else but. Of course, antivenoms won't stay clear of that forever. So I think it's actually quite urgent that we have these alternatives. Like David said, eventually we will get shut down and we won't have any way to validate that these products are safe and effective and should be out in the public, per day. So I think it's really important that we do actually come up with alternatives as quickly as we can, and that we should be collaborating to try and find ways to validate alternatives and get them into pharmacopoeias so that manufacturers have those alternatives available to them. But from the audience, I think if we could just, anybody has questions for the panel or things that they'd like to comment on, there's a few manufacturers here. They may have some comments they'd like to make, so please go ahead. Sale.
I'm just curious, in the spirit of refinement, there's quite a bit of instrumentation one can outfit an individual animal with. I'm curious what your thoughts are collectively on the types and availability and variety of instrumentation available for small animal models. And do you see that as being sufficient or insufficient for being able to reduce and refine the animal work that I think pretty clearly needs to be done? But by being able to get more data out of each animal, would that lend itself to production or better model, if you will?
I mean, maybe I'm not so creative, but most of the animal studies that we do is the mouse dead or is it alive? It requires limited instrumentation to see if it's stiff. But I guess for some of the more, if you want to sort of study effects, not so much for quality control, but more to sort of see, is your new molecule having the right effect on local tissue damage? Then my guess is you can probably get more out of each animal. You can use more advanced proteomics as an example to go in and actually really try to understand what's going on. This is maybe less relevant for existing antivenoms, but more important for completely new modalities that might not be toxin neutralizers, but might be tissue regenerating factors and things like that. So I think for research, probably yes, but right now it's often super simple endpoints, life and death.
I think that's a very interesting point, because monitoring even free roaming experimental animals for things like blood pressure has become possible. And I think another major problem about the mouse model or the rodent model is it a misleading model? It may be the best we've got. There are situations clinically where it gives a very misleading answer. And I think, as you said, it's a difference between a living and a dead mouse is a rather unsubtle way of looking at the complex pathophysiological processes which, with different venoms in the human model, cause very different effects. And a particularly gross example, I've already been discussing this with you, Anne, is that the spitting cobra venoms, which kill mice very quickly in sort of convulsions, but in a human very rarely kill them, but lead to incredibly common tissue damage and complications of morbidity rather than mortality. So a gross example of where the mouse is not a useful model for the human. So I think that your idea would be well worth pursuing, and as you say, it would reduce the number of animals involved, but it would increase the amount of information in each of the chosen animals.
Okay, and then we've got a question here as soon as Juan's done.
Yes. Instruments they are, of course, and you can measure the temperature and make a correlation between the time to die with the temperature, with change in the temperature. You can measure the metabolic rate, the respiration, etc. The question is not, you will always make the correlation, but is this correlation correct, or is it useful to translate to units that you can use for in research that is focused on humans? So this is the question. Most of the alternative methods are qualitative, but are not quantitative in a way that you can reliable translate to a number that says like an LD50, this amount of venom kills 50% of the population, or this amount of antivenom has a potency of neutralization, this amount of venom. So this is one of the problems. The other with the refinement, we are forced by our committee, ethics committee, to observe the animals 8 hours a day and take decision on whether we need to euthanize or not the animals. We also are committed to use analgesics. We get to a compromise that we will not put 100% animals with analgesics, but some sentinels in each experiment to see if analgesic has any effect on the outcome of the statistics of the animals. And our conclusion is that for some venoms there is no interference at all. Some others potentiate or make lower the toxicity of the venom, and some others they have a clear effect but not reproducible in all the animals. So at the end, our conclusion with using this compromise with the ethical committee is that we have used more animals that we had to use with if we had not put sentinels in the different experiments. So on the time to die, in the observation, I asked the lady that he said, how are the animals doing? And say, there are 2 that are so tomorrow they will die. And tomorrow they were like you had never injected venom on them. But this is the same. Probably you, David, would tell a bitten person not to move, so to maintain the venom without in the circulation, not to exacerbate the distribution of the venom. And the animals do the same. They stay something like this, without moving, without breathing fast, and some of them survive and some others do not survive, but you cannot predict it. So if I had give permission to euthanize all these animals, the statistics, the ones that we have the tables with, which one was in this situation and what was the next day and when they died, etc., then would be absolutely different in many cases, in some others not. But it's unpredictable which ones would be in these statistics and which ones would be on the other. So we try to do our best also with three R's, but at the end his result to be negative in terms of using more or less animals or refine a process.
Just make a point. I mean, sorry, can I just? I think the point about endpoints is great because I guess the question is, do we have to let an animal get to that point? Does that have to be an endpoint? If the animal is that severely envenomed, where it may survive or it may not survive the next day, why do we have to be constrained to take it to a 24-hour time point? That's just convention, right? So I think we need to develop much better endpoints, and as you say, there are ways now to monitor different parameters. The challenging thing we've got with snake venoms is the diversity, right? And the diversity of pathology we see. So we currently use lethality as our endpoint. Yes, no, die, not. And it's very clear cut. It doesn't matter kind of whether you get a systemic neurotoxic effect or a hemotoxic
And diversity in the animals. There is always a super mouse that never dies. But you cannot looking at the eyes of the animal who is this super. If this were the case, you will not use this because it will break your statistics.
Yeah, Rebecca.
But it may well be we need different endpoints for different types of envenoming, and that will consume mice, which is kind of the point you made, Juan.
Well, I mean, from a WHO perspective, with our testing for Nigricollis, for example, the necrotic and hemorrhagic myotoxic venoms, we tend not to rely so much on ED50 as on, the MHD or the MMD or the other alternative tests that actually look at the ability of the antivenom to neutralise that specific toxic activity itself. And we find that sometimes the antivenoms are much better at neutralising those specific activities than if we were to just rely on an ED50. So Nigricollis, I mean, they die. That's no problem. But people very rarely die from blackneck spitting cobra bites. But when you look at the dose of effective antivenom for the specific activity, whether it's the necrotic or the myotoxic activity, you get a very different result. And it's probably much more reliable. But Rebecca has a question, I think.
Yeah, so now we're just going back to your equipment to get more data from the same animals. So Stuart's first group now have been using point of care testing, so they were able, maybe more Amy and Stuart, were able to refine down exosolates to the point where they had an assay that was measuring an effect on fibrinogen while seeing no evidence of envenoming in the mice, and then having a window where you can measure therapeutics in. So that sort of equipment definitely exists. We're now looking at it for neurotoxicity. Stu's bought a new bit of kit that will measure sort of respiration rate as a proxy. So it all just takes time, but there's definitely the equipment out there that you can refine a model to not have to be, is the mouse alive or dead? Because lots of antivenoms are determined good on the basis that a mouse alive looks absolutely horrendous. And so that's not really representative of a great treatment.
I think we are too much in agreement, so I'm going to throw in a bit more of a statement that will probably divide the waters a bit more. And before everyone runs off and spends all their time on making fancy in vitro models and saving all the mice lives, shouldn't people spend more time on actually improving antivenom and making it into either synthetic or recombinant product, where you would save many, many, many animals for centuries?
Of course, I agree with Andreas. I said that in my first moment. I hope we can do that pretty soon, and I think you are responsible for that partially. But I want to say a little bit about the second arm, the reduction, and taking what you were talking about that. How many mice we are killing just trying to do LD50s and ED50s in venoms that don't kill. For example, if you take Botrops venoms, they don't kill. It's just morbidity. So even WHO still recommends ED50s, LD50s. So I think we have to change our minds about that. Okay, it's a bit what Nick said as well, but we need to push hard
And this is where we are, I guess, because the the ED50 test is in the pharmacopoeia, and that's what
Yeah,
the
but
regulators rely on.
We have to change it.
Our guidelines have all the other alternatives, but so far none of the regulators have picked them up because there's no validation data for them. And to give you an idea, where there was a model antivenom and a reference venom established in for North Korea, China and Japan for their mamushi antivenom. They had to validate the reliability of the potency testing of the venom as well as the effectiveness of the antivenom against the specific activities in collaboration with, I think, 12 independent national control laboratories. Their regulators wouldn't accept academic laboratories. They had to be done by, you know, the Australian TGA, the US FDA. They had to be done by major international reference laboratories that had full accreditation. And that makes it all the more difficult, to be perfectly honest. David.
Sorry, I just want to dispel any idea that Andreas is correct that we're in agreement. We are not in agreement. What I'm hearing this afternoon amounts to an unacceptable amount of animal suffering in the interests of a not particularly well-defined endpoint of relevance, therapeutic relevance. And I think it's no good being complacent like this. More has got to be done. I know that some people are looking for alternatives. I mean, Paula Sells produced one of the earlier ones, the chick chorioelentois. But clearly the range of activity that these alternatives have come up with is extremely limited. So I would say an unacceptable level of suffering in these laboratory mice, an unacceptable and I think irresponsible reluctance to use analgesia, and thirdly, an inability to see how imperfect the mouse is as an in vivo model for human envenoming and antivenoming treatment. So I just want to stir up the argument a bit. We are not in agreement, Andreas.
It's still too much in agreement, I think so.
Okay, here we go. I have a question down the back here. So we need to have a process. Now, WHO's guidelines for antivenom production are being revised in time to go to the ECBS next October 2026. There's an opportunity, if the community is interested, to look at those chapters that relate to the use of animals. I mean, in the last edition we sort of strengthened it by introducing the 3R principles and all of that. But in terms of the testing, again, it still largely follows, well, ED50s are what you go with. We need to go a bit further. We need to provide a basis for the validation of the alternatives and lock that into the guideline. And then we need to sort of give some priority to which alternatives should be favoured. And encase that in the guideline as well, when that goes to the WHO's expert committee, if they adopt that, it'll be published in the technical report series, which will go to regulators all over the world, which will make it a lot easier for, say, VINs or Premium or Microfoam or whoever to actually be able to put a proposal forward to say we want to use this as an alternative and it's recommended by WHO. So that would be one way to do it. So when we start the editing process in the end of this year, early next year, putting that on the agenda and getting a group of people who are willing to contribute to it might be a way forward. Any other questions or comments? Yeah, Siddharth.
Yeah. Very interesting panel discussion to have. We actually started, working with some potency assays on the non-animal model. And we had a presentation about a few years back on AI generated model. So just wanted to share that, preliminary results and the proof of concept were very good. But during the project took us about 1 year's time. What we realized is for AI to be very successful, we required a huge library of information in order for it to be more accurate. So would be happy to discuss with any scientist or anyone who is willing to, have interest in taking up this project. That's one. Second, it's not for manufacturers, like as Dr. David said, that it has to be a validated method in California for us to implement. Happy to do that. But at least for the pyrogen test, there is a validated method which is called the MAC. If that can be probably, in the next technical series published, then at least for the pyrogen will be a starting point where we'll be using a non-animal test for, doing it successfully. So that's just a suggestion.
One.
I have a comment that I disagree that ED50 is unnecessary, because the ED50 is not only a test for measuring how an antivenom can save 50% of the population or 100% in potency terms. It's an assay that reveals a property that is intrinsic to the antivenom, which is its capacity to bind and neutralize toxins in a quantitative manner. And this, if you combine with in vitro tests, like we developed antivenomics, which is an in vitro test for determining the binding capacity, toxin resolve binding capacity against the venom. And when you combine these two in vitro and in vivo testing, then you can also calculate easily what's the percentage of active product in the antivenom, which toxins are well recognized and neutralized. If you combine with toxicovenomics, which is the same as ED50, but for each of the toxin, etc. So it's an amount of information about the antivenom itself, intrinsic properties, its intrinsic potency, and that you can compare with other products of the same antivenom or different antivenom, that is so valuable, but can only be calculated with combining in vivo and in vitro analysis. So it's basic research in a sense, but it provides data that are very valuable for translational research.
Okay.
If you like. And I think Juan's just made a really good point that we perhaps haven't discussed as much as we should have done, which is particularly when we're developing and trying to test new approaches or new products or new antivenoms, whatever they might be, that in vitro testing piece has to be performed quite rigorously and in a diverse range of experiments. Yes, we don't have a single assay that can replace an in vivo model, but we do have lots of different assays that can help guide and kick out things that shouldn't be ultimately tested in an in vivo model. So we need to use those filters really robustly. And I guess the final point I want to make is that we shouldn't lose sight of the fact that the model we use now, which is the regulatory approved model, as you said, David, it is imperfect. It doesn't reflect envenom read accurately. It doesn't mimic how things are dosed. It does enable us to do those potency comparisons, but we're already relying on an imperfect model. So I think there's an even stronger impetus on us to develop alternatives to ultimately replace it.
David Worrell, do you want to close this up with just a last comment?
I'm not sure that's fair in such a bitter argument, but
well, I know you love the last words.
I think Nick, I think I would agree with Nick. I think he summed up. I mean, the existing model is not a perfect one, and people who adhere to it at all costs should not be blind to the fact that it is a very imperfect model. I think that if this discussion has in any way aggravated, provoked, or stimulated toxinologists in the audience, at least to look a little more critically at their use of in vivo models, such as the mouse model, then I think that would have been a good thing. I think reducing, mitigating suffering until we can completely rid ourselves of the need to experiment on animals should be a priority. It shouldn't just be a sort of luxury, a cosmetic addition. I think it should be central to our use of animals that we should try and reduce their suffering while accepting that they don't provide an adequate model for human envenoming.
Great. And on that note, I'll thank the panel very much. Thank you all, and we'll move on.
At the end, we need to develop artificial mice which have plenty of sensors to sense a parameter that has to do with each of the functions. And when you inject the venom, then see how the panel of sensors change and how you can put back to the normal.
They'll breathe and they'll take in the well.
Because if this were so, why do you put 5 LD50s into a mouse? You know all of them will die. Don't do the experiment. But if you don't have this data, you can do the statistics. Yeah. They will put cures back.